transcription occurs inside of the nucleus. First part of transcription is initiation. During initiation, RNA polymerase binds to the DNA strand. For eukaryotes, promoters must be accompanied by transcription factors in order for RNA polymerase II to bind to the DNA strand. Second part of transcription is elongation. During elongation, RNA polymerase continues to unwind the DNA downstream-wise. RNA elongates as new nucleotides are added onto the end. Rather than thymine, adenine will correspond with uracil instead. The third part of transcription is termination. During termination, RNA transcript gets released and the RNA polymerase II attaches.
During transcription, a methylated cap made of guanine and 3 binding phosphates is attached to the 5' side. This cap helps to prevent degradation by exonucleases and protect the mRNA. On the 3' side, RNA polymerase synthesizes the poly A tail, which is made up of a lot of adenine. This leads the mRNA outside of the nucleus and into the cytoplasm.
A small ribosomal subunit then attaches to the binding site of the mature mRNA at the 5' cap. The first tRNA carrying the thymine arrives to the A site and attaches its anticodon to the corresponding codon on the mRNA. This tRNA then moves to the P site. After this first tRNA has moved to the P site, a large ribosomal subunit attaches to complete the ribosomal complex and initiation of translation. Each amino acid attaches to a specific tRNA via the enzyme, aminoacetyl tRNA synthetase. It attaches through ATP. The ready tRNAs go into the A site of the ribosomal complex and attaches its anticodon to the codon on the mRNA strand. The tRNA in the P site transfers the growing polypeptide to the tRNA in the A site, adding on the next amino acid from the tRNA out. After, the tRNA in the P site exits the ribosome through the E site where it can go back to aminoacetyl tRNA synthetase to be reused. The tRNA in the A site with the polypeptide then goes to the P site. This process repeats until a stop codon signals the end of the amino acid chain. When a release factor enters the A site, it signals for the polypeptide to be released from the ribosome as translation is complete. The mRNA and ribosomal complex disassemble into the cytoplasm as they wait for another mRNA to translate.